The overall goal of this procedure is to introduce a fluorescent unnatural amino acid into a membrane protein expressed in Xenopus oocytes, and to determine the protein's function and structural rearrangements simultaneously using voltage-clamp fluorometry.
This technique will help answer key questions in the field of structure-function relations of membrane proteins. Using voltage-clamp fluorometry, we can directly correlate structural rearrangements with protein function. Adding fluorescent unnatural amino acid labeling to it helped to overcome previous limitations on the labeling. This was once the accessibility of the labeling sites as well as background labeling due to endogenous binding sites.
The implication of this technique extends towards a better biophysical understanding of membrane proteins because you can now probe domains which previously were inaccessible with traditional voltage-clamp fluorometry.
After surgical removal of the Xenopus oocytes, wash the oocytes 3 times with SOS solution. Select large and healthy oocytes individually and incubate them at 18 degrees Celsius for at least 4 hours in Barth's solution supplemented with antibiotics and 5% horse serum before injection. For nuclear injection of DNA, prepare a long and thin injection tip to reach the nucleus without damaging the oocytes. Then, fill the injection tip with oil and mount the injection tip on the nanoinjector device. Next, install the nanoinjector under a stereo microscope and break the end of the tip with forceps. After that, eject the oil until there is no air bubble trapped inside the end of the tip.